Yeah, I think let's slowly admit people from the waiting room into the meeting.
Yeah, so we are live now, and yeah, we are good to go. We can start.
Oh, okay. Yeah, I think let's slowly admit people from the waiting room into the meeting.
Yeah, so we are live now, and Dr. Vatsala also joined. Should
Oh,
I
fantastic.
all? Yeah.
Yeah,
So
we
I'm
are good to go. We can start. Oh, okay. Okay. Sanjana, if you can start the video. Hello, everyone. Sundaram, if you can please share the slides. Welcome, a very, very warm welcome to you to this session of Her Story of Science, celebrating women in research. We are Biologically Speaking, and we are organizing this conference in association with Bias Watch India. Thank you all for joining from all over the world. We are very grateful to you for this continued support. We are commemorating the International Women's Day of 2021 by bringing to you this two-day conference consisting of 7 sessions, each dedicated to a woman scientist and 2 keynote lectures. This is the Veronica Rodrigues session on neurobiology. Veronica Rodrigues was born in 1953 in Nairobi, in Kenya, to Indian parents from the state of Goa. After completing her schooling in Kenya, she initially enrolled at the Makerere University in Kampala in Uganda, but she had to quit because of an outbreak of political unrest. She then won a scholarship and moved to Trinity College Dublin to complete her BA in microbiology. Inspired by a paper in the Journal of Molecular Biology by Dr. Obaid Siddiqui, she first came to India in 1977 to pursue her PhD at the Tata Institute of Fundamental Research in Mumbai under the guidance of Dr. Siddiqui, and contributed immensely to the field of olfactory behavior in fruit flies. She then moved to Germany at the Max Planck Institute in Tübingen to obtain her postdoctoral training under the mentorship of Karl Keats and Erich Buchner. In landmark experiments conducted during her postdoctoral studies, she uncovered how olfactory information is encoded in the brain. She later returned to TIFR to continue her research on neurobiology and became a fellow of the Indian National Science Academy. Her lab focused on understanding the link between gene expression, cellular communication in nerve cells, and the behavior of an animal. She later served as professor at the National Center for Biological Sciences in Bangalore. Apart from the illustrious lists of awards and honors, Veronica was instrumental in creating and maintaining rich collaborations with scientists, both Indian and international, and her contributions were absolutely crucial in setting up the neurobiology climate of India. She breathed her last in 2010 after a 5-year-long battle with breast cancer. Biologically speaking, remembers and honors the true internationalist, the pioneering neurobiologist with a spirit of adventure, and brings to you the Veronica Rodrigues session of neurobiology, featuring two fantastic women neuroscientists of our time, Dr. Vidita Vaidya and Dr. Vatsala Thirumalai. Our first speaker for this session is Professor Vidita Vaidya. Professor Vidita Vaidya received her undergraduate degree from St. Xavier's College in Mumbai in the Life Sciences and Biochemistry. She obtained her doctoral degree in neuroscience at Yale University in the lab of Dr. Ronald Duman. Her postdoctoral work was done at the lab of Dr. Ernest Arenas at the Karolinska Institute in Sweden and Dr. David Graham Smith's lab at the University of Oxford in the UK. She joined the Department of Biological Sciences, TIFR, in March 2000 and has been a Wellcome Trust Overseas Senior Research Fellow and an INSA Fellow from 2015, and has recently been inducted as a Fellow of the Indian Academy of Sciences in 2021. She received the National Bioscientist Award in 2012 and the Shanti Swarup Bhatnagar Award in the Medical Sciences in 2015. In 2019, Professor Vaidya won the Nature Award for mentoring in science as a mid-career mentor. She's also very active in science outreach efforts. She's been featured on the Life of Science blog, has 2 TED Talks to her name, and has also been featured on the Chai and Why series of outreach talks conducted by TIFR, and many other such initiatives. And she has also been very vocal about gender equity issues in academia. Her research at TIFR focuses on the neurocircuitry of emotion, its molecular signatures, and the changes that they undergo through experience or through psychiatric disorders. Please join me in welcoming Dr. Vaidya to Her Story of Science. Over to you, Dr. Vaidya.
Thank you, Shreya. It's a pleasure to be here and to be part of this wonderful 2-day meeting. I'm also particularly honored that I get to be part of the Veronica Rodrigues session, because I count Veronica as both a mentor and a dear friend. So it's absolutely my honor to be able to talk in this particular session. So I'm going to talk to you today about a serendipitous discovery we made a few years ago. And it's going to be titled Serendipity and Serotonin, and I'll tell you about that. But first, I want to really celebrate Veronica. The Department of Biological Sciences at TIFR carries the marks of her personality in many ways. Remarkably generous as a scientist. And this is everyone who joined the sort of department when she was the department chair will attest to this, that her freezers, her microscopes, everything was just available and easily accessed. And her personality is captured here in these photographs, starting with when she was a graduate student doing a tug of war, to the we were much more used to seeing, which is chatting with a drink in her hand or a cup of coffee, where she would, discuss science with you and we would get into all sorts of debates. But I wanted to actually share this letter she wrote, because I think it's appropriate in this context to share this letter that Veronica wrote. Now, this is a letter written in 1990, where she was writing to the then Dean R. Vijayaraghavan, where she was raising a very, it's a very polite and extremely firm letter, where she writes about why our women faculty at TIFR addressed as Kumari or Srimati or Ms., if the equivalent terms are not also applied to the preponderantly large male faculty at the TIFR campus. And this in many ways just captures her personality, because that is who she was. She would call a spade a spade. And she was tough, unrelenting, but also remarkably supportive of young faculty. And I've been very lucky to count her as my mentor and also as a dear friend. So this is actually a paper that Veronica wrote and was published the year that she passed away. It was work done by Ajit Pratap Singh, her graduate student, in collaboration with Vijayaraghavan, who was one of her close collaborators at NCBS. And this is really, the reason I put this up is because this is a serotonin neuron. Okay, this is a serotonin neuron in the fly brain. And Veronica stumbled on this particular really interesting neuron, which undergoes major architectural remodeling during olfactory development. And she discovered essentially that neuronal activity and Wnt signaling were essential to the pruning of this particular neuron when it eventually takes on its mature final phenotype in the adult fly brain. And Veronica and I would often discuss serotonin, and I think she would have enjoyed this work that we published recently. And so this is in honor of her amazing spirit for science that we're celebrating her today as well. Okay, so let's segue into talking about serotonin. So serotonin is an ancient chemical. It's a small molecule. It's derived from all the tryptophan that you eat. So, the serotonin that you make in your body comes from the tryptophan that you consume. And then that tryptophan is hydroxylated and then goes through a amino acid decarboxylase step, and then eventually gives you 5-hydroxytryptamine, which is basically serotonin. Well, most of us, when we think about serotonin, will immediately think about regulation of mood, regulation of behavior, regulation of anxiety and depression, partly because that's what we associate with this neurotransmitter. But what I want to bring you back to is the awareness that this neurotransmitter existed when there were no neurons. So if you think about the evolution of this particular molecule, it's really ancient. It's a really ancient molecule. It's been around for about 3 billion years. Right from the very earliest stages of multicellularity. It's there long before anything that even remotely resembles a brain shows up. It's there long before receptors for serotonin show up. They show up like a good almost 1 billion years later. So it's really quite remarkable that there's this molecule that's floating around. It's abundantly present in plants. So this is really interesting to think about. When we traditionally think about serotonin as neurobiologists, we're always biased to thinking that it's the critical player in the brain. But what was it doing long before it ever got co-opted in the nervous system? So there's been evidence about this for a long time. In the literature, people have talked about how it modulates cellular activity. It may actually act as an antioxidant. It's almost a trophic factor. There is old data that indicates that when you cut a leaf, the leaf browns, and anyone who's a bad nurturer of plants would have done this, where you're trying to, prune your plant, but you actually end up damaging the leaf and the leaf is browning. Turns out if you apply serotonin to the leaf, then it'll prevent that kind of browning. And it's almost been debated to work almost like an antioxidant. So this molecule is really interesting. It's very abundantly present in primitive metazoans. In planaria, it's implicated in regeneration. In tetrahymena, it regulates phagocytosis. So why am I bringing all this up? Just to tell you that it was there long before nervous systems existed, and it might have functions that are conserved and earlier than its neurotransmitter-like effects in a sense. And that's of interest to many of us. So to make serotonin, you need oxygen around. It undergoes a critical rate-limiting step where tryptophan goes through tryptophan hydroxylation to produce 5-hydroxytryptophan, which then eventually produces 5-hydroxytryptamine, which is serotonin, the final molecule. So you need oxygen around. And when you think about who's making oxygen, obviously these are algae on the left, and you can think about plants. You automatically think about plants as being the source of where you can get your own oxygen eventually. And plants evolved, of course, the critical intracellular organelle, which is the chloroplast, and that not only captures light, but also is a source of rich tryptophan synthesis. By the way, along the way in animal evolution, we lost the ability to make our own, tryptophan. We have to now eat it. I mean, all the tryptophan we get has to come as an essential amino acid from whatever it is that we consume, but plants make their own tryptophan substantially. And tryptophan is fascinating, not only because as an amino acid, it's a component of many of the chromophores that absorb light, but it's also an interesting precursor molecule to many other interesting chemicals that then have diverse biological effects. So there's an interesting hypothesis, and this idea had been floating around for a while, and been debated, discussed, but not really pursued. And it's an old idea. It was around all the way back to the 70s, where Ephraim Asmitia and others had suggested that, as you develop the photosynthetic ability, essentially, to make your own food, which is what evolved in plants, you also generate free radical species in the process of that reaction itself. And those free radical species will need to be scavenged, because over a period of time, otherwise you will accumulate free radical oxidant damage in these cells. So that idea then requires you to also think about the co-evolution of something that would act as a potent antioxidant. And it was debated that maybe serotonin and the ability to synthesize serotonin, which is also derived from tryptophan, would then be a critical component of perhaps having antioxidant-like effects. And that idea is old. There was some evidence for, against, but it never really got pursued very actively. And it is true that our understanding of what serotonin does got sort of subsumed with the neurotransmitter identity of serotonin. And at least for neurobiologists, that's what we've paid large amount of attention to. But we've paid far less attention to these conserved roles of serotonin that may be around and have been around, and certainly were around in systems where there are no neurons, but may also be conserved within the nervous system and the brain. And that's what I'm going to tell you about today. So really the question is, does it do in the brain what it originally started off doing? I mean, does it have this sort of a neuroprotective, antioxidant, mitochondrial modulatory effect, which is an ancient, conserved, evolutionarily conserved effect of serotonin? And that's the idea behind the question. So we sort of stumbled on this. In fact, we completely stumbled on it. Turns out we were actually interested originally in looking at neuroprotective molecules that are plant-derived, and we were looking extensively at plant-derived, plant-derived actives and psychoactives, and we were carrying out quite a bit of research, which was at that point in time sponsored by industry, and we were, collaborating away between a few labs where we were looking at this, and we needed a control, a positive control. So growth factors became a very natural positive control. But when you're a behaviorist who's interested in serotonin and your entire lab focuses on serotonin, it's almost as though you can't resist the temptation to dump serotonin into every little project that you have. And that's exactly what happened here. The postdoc who was working on this project said, what if I also look at serotonin? And then that's where we digressed, and we only ended up looking at serotonin and gave up the original project because we stumbled on something that was quite interesting. But now when you think about it, it seems almost so intuitive and natural that serotonin, which is a potent regulator of neuronal plasticity, growth, differentiation, aging, mood, behavior, may also impact the very molecule, the organelle, the mitochondrion, which regulates cellular energetics and is also a critical component of mediating plasticity effects in the brain, modulating growth and modulating aging. It seems intuitive, but it wasn't at the beginning when we started, where we just actually stumbled on it purely by fluke. So we were interested in asking, does serotonin regulate mitochondrial biogenesis and function? And I'd like to acknowledge the people who did the work. Sashena Fanibanda was a very talented postdoctoral fellow in the lab at that point in time, and is now assistant professor and works closely with my group. My collaborator, Ullas Koltur, in the Department of Biological Sciences. I have the rare privilege of working with my father, Ashok Vaidya, on this particular project. Babu Krishna and Sukrita Babu Krishna is a graduate student from Ullas's lab, and Sukrita is a former master's student of mine. And Noelia and Jay, who very generously shared some critical mouse lines for our work. And this is a team of people who really worked on this. So what did we set out to ask? We first set out to ask, okay, if serotonin indeed has this conserved broad role where it's actually targeting bioenergetics, does it impinge on mitochondria? In the first, most basic sense, does it actually contribute to the generation of new mitochondria? And that's what we set about to ask. So we grew neurons, and this is Sashena's work, where she grew cortical neurons from the rat brain, also from the mouse brain, but I'm showing you data from the rat brain here. So you grow them from the E18.5 cortex, you grow them in vitro, and at 7 days in vitro, you apply serotonin into the medium. And we've done this across a wide range of doses for a wide duration period. And I'm just showing you some of that data. What you can clearly see visually, it's already obvious that you end up with higher mitochondrial intensity, at least based on the staining for a mitotracker dye that labels mitochondria. And you can quantify that. It's a semi-quantitative approach to look at whether you end up with more mitochondrial mass. But that's a proxy measure. And so you want other ways of showing the same thing. And what you have here is staining for VDAC, which is an anion channel present on the mitochondrial membrane, and is again a measure of how many mitochondria you have. If you have greater expression, it would indicate, but not categorically show you, but strongly indicate that you end up with more mitochondrial mass, and that's why you have more VDAC expression. And that's what we see. that you see an enhanced intensity of VDAC expression, both from immunofluorescence as well as from Western blotting, that you can clearly see that across the doses of serotonin, you end up with more mitochondria. But a better and more direct measure of this is to actually quantify the mitochondrial DNA content, which is perhaps the closest measure you have of asking, do you have more mitochondria? And so across the dose range, what you can see is that when you bath apply serotonin to cortical neurons, and it is cortical neurons, you don't see this effect with hippocampal neurons, you don't see this with muscle cells. We haven't looked at epithelial cells, and that's an area that will be very interesting, will end up showing you an increase in mitochondrial mass, here measured by looking at the amount of mitochondrial DNA content. In the interest of time, I'm not going to tell you how we dissected out the entire pathway that mediates it. Suffice it to say that the master modulators of mitochondrial biogenesis, SIRT1, SIRT1, and PGC1 alpha, which is a transcriptional coactivator, are absolutely essential to this process. And if you do not allow SIRT1 to function either catalytically by blocking it with a catalytic inhibitor or by actually using knockouts, you will lose the effects of serotonin. So that's just telling you that we know the pathway that's involved, and that's not very surprising. This is a conserved pathway that regulates mitochondria on multiple different cell types, also in the brain. And it appears to be that somehow serotonin is sitting upstream of this pathway, recruiting this pathway, and resulting in more mitochondrial mass. So it would be good then to really look at mitochondrial function as well and ask, okay, if you have more mitochondria, do you end up with more mitochondrial function? Now, there's two ways of thinking about it. If you have more mitochondria, automatically you would presume you would have enhanced function. That doesn't tell you whether you have more bang value per mitochondria. It simply tells you because you have more mitochondria, you have more function. What we went on to show was that you actually also end up per mitochondria enhancing ETC chain coupling likely and enhancing efficiency per mitochondrion, and that I'll show you in a minute. So the bottom line on this, looking at cellular ATP levels, as expected, if you have more mitochondria, you end up with more ATP production. Not very surprising, kind of intuitive, kind of expected. But what about if you isolate mitochondria and actually start looking at this more carefully? And we've done that as well. But here you see in cortical cultures that if you actually measure basal and maximal respiration as well as ATP coupled respiration, neurons that have been bathed with serotonin will give you enhanced maximal respiration, basal respiration, ATP coupled respiration, and also end up with a substantial increase in spare respiratory capacity, which strongly tells you that these cells that have seen serotonin for a period of time are almost as though they are primed to function at their optimal mitochondrial output capacity, and under stress and strain, they may be actually far better able to cope. And there I'll give you some data that strongly indicates that. So we know that serotonin is doing this, but how is it doing it? Serotonin has something like 16 receptors, all of which are present within the cortex. Pretty much most of them are present in cortical neurons. So how do you even begin to start to tease this apart? We were sort of lucky. We stumbled on the receptors very quickly in our hunting. We went through the 5-HT1A, 5-HT2A. Very early on, we figured out it was the 5-HT2A receptor, and that's what I'm going to show you next. So here you have neurons grown in vitro. After they're grown in vitro, you come in with an inhibitor of the specific 5-HT receptor. In this case, I'm showing you data from the 5-HT2A receptor, which is abundantly expressed in cortical neurons. And then you also have, obviously, serotonin on board, and you ask, will serotonin work if you don't allow it to hit the 5-HT2A receptor? And here's what you see. You can see this is the data with the relative mitochondrial DNA content on the top. And this is, again, data when you stimulate the 5-HT2A receptor. Serotonin, as expected, gives you a robust increase in mitochondrial biogenesis. That effect is completely lost if you use a pharmacological inhibitor of the 5-HT2A receptor, MDL100907, totally blocked. If you stimulate the receptor in contrast, you actually increase biogenesis. And that's also true for ATP. ATP production goes up with serotonin, blocked by the 2A receptor. If you stimulate the 2A receptor, you stimulate function and biogenesis. This is also true when you use knockouts. So we have a wonderful line, which is a rescuable line. You can actually knock out the receptor, and because it has a floxed base rescue cassette, you can actually flox out the cassette and bring back expression back to normal. So here are wild type neurons. Here's the 5-HT2A receptor expression. Knockout, you lose the receptor. In a rescue, Cree-based rescue strategy, you can actually put back the receptor. And when you do that, What you see is the effects of serotonin are there in the wild type neurons, lost in the knockout, and restored in the strategy where you restore back expression, strongly implicating the 2A as a critical component of this phenotype. That's also true for ATP. So again, in the interest of time, I'm not going to show you all the blocker experiments that we did, but we figured out the pathway. Serotonin through the 5-HT2A receptor, via recruitment of phospholipase C and MAP kinase, via SIRT1 and PGC1 alpha, strongly drives a mitochondrial biogenesis program, which eventually results in increased mitochondrial mass as well as enhanced function. Now, this is interesting, but all of this that I've shown you so far was done in a dish with neurons grown in the most artificial conditions as possible. They're primary neurons, but they're grown on a plate. It's not exactly how neurons actually are. They're in the brain, surrounded by a whole bunch of astrocytes, other things, etc. So we wanted to actually ask, is this also true within the nervous system? And we use a surgical approach where we put osmotic mini pumps, which we then allowed us to directly infuse serotonin straight into the brain. And when we do that and look within the cortex, we get an increase in mitochondrial DNA content as well as in ATP, strongly indicating that this is an effect that's seen both in vivo and in vitro. We then used a bigenic mouse line to drive activity of serotonin neurons, endogenous serotonin neurons, driving them to release serotonin. So this is a PET1 Cre HM3D cube mouse line, which is a chemogenetic mouse line. It allows us to selectively drive this chemogenetic receptor, which will allow us to activate these neurons using an exogenous ligand, only in PET1 Cre neurons, which are the serotonin neurons of the raphe, as well as other parts of the brain, but mainly in the raphe. What we can then do is stimulate these cells to actually release serotonin. So now instead of giving serotonin, we are driving the brain to release its own serotonin. And when we do that, we also see an increase in mitochondrial DNA content, ATP, and several other measures of mitochondrial mass, strongly indicating that whether you pump in serotonin from outside, you increase it pharmacologically, or you drive the system to make its own serotonin and release it, you will end up with enhanced mitochondrial biogenesis and function in the cortex. I'm going to skip this because I'd rather spend more time talking about, have time for questions. So bottom line, the same thing happens if you stimulate just the 5-HT2A receptor. So what have I shown you so far? I've shown you that serotonin through the 2A receptor recruits a very well-conserved pathway of SIRT1 and PGC1 alpha to drive NRF expression, etc., to eventually drive a program of mitochondrial biogenesis. This seems to be rather unique to the cortex in that we don't see it in other cortical structures like the hippocampus, likely because of the nature of distribution of receptors. The 2A receptor, which is a GQ-coupled, phospholipase C-coupled receptors, abundantly present in your cortex. So you're driving more mitochondria, you're enhancing function. All of this then begs the question, what is the point of doing this? I mean, why would you have the system where a neurotransmitter classically associated with modulation of neuronal firing and behavior is now working in a more sort of almost like a hormone which bathes the brain, and serotonin is really released in both a phasic and a tonic manner. So bathing the brain with serotonin somehow seems to be sufficient to drive a bioenergetics program within these critical circuits. What might this be doing? And so what Sashena set out to ask is, does it matter for how cells then survive in the brain? And what she did is she actually looked at ROS. And so what is interesting is serotonin itself reduces ROS dramatically, likely because it produces higher levels of SOD2 and catalase, dramatically upregulated when the neurons are bathed with serotonin. And then when you try to kill the neurons or damage the neurons with conditions like kainate or hydrogen peroxide, which is classical oxidant damage, you will end up, of course, producing a lot of ROS, but this gets scavenged if these neurons have actually seen serotonin before. And that's reflected as well here in the cell death curve. And let me just show you, focus on the image on the right. That's perhaps the most clear demonstration of it. So across a very wide range of excitotoxic kainate doses, which would kill neurons, and as expected, it does. So, I mean, here's 100% viability. Neurons are dying if they are exposed to this excitotoxin. But if the neurons have been bathed with serotonin before you come in with an excitotoxic factor, look at the wide dose range of kainate across which serotonin will protect these neurons, potent neuroprotection. But this neuroprotection requires SIRT1 activity, because if you use a catalytic inhibitor of SIRT1, you lose all the protective effects of serotonin, strongly suggesting this is likely through a mitochondrial mechanism. We also know this from SIRT1 knockouts. In SIRT1 knockouts, all the neuroprotective effects via serotonin are lost. And that again implicates this pathway as being central to the protective effects that are mediated. So here's our working hypothesis. And this was really a finding which I think Veronica would have enjoyed thoroughly, because truly we stumbled on it. We weren't looking for it. We were just doing something else. And we happened to find that serotonin, which is monoamine neurotransmitter critical for stress modulation and stress resilience, has this fundamental pathway it seems to impinge on, which might directly regulate how neurons buffer stress. So what it does is, through the 5-HT2A receptor, through a very well-conserved pathway, drives mitochondrial biogenesis, reduces ROS, enhances bioenergetics and coupling, increases oxygen consumption, and collectively results in a state in the neuron where the neuron is neuroprotected and has a enhanced buffering capacity. And that's fascinating, because at the sort of whole animal level, the fact that serotonin is a big contributor to helping animals buffer and handle stress is something that's a really well studied and well understood idea. But at the cellular level, where the stress resilience may be directly modulated through some sort of a bioenergetic pathway, is interesting, novel, and opens up a whole bunch of interesting questions to ask. So I'm going to wrap up by acknowledging Sashena, Ullas, my dad, who I work with. This is my amazing team, who I'm very privileged to work with every day. Jay and Noelia for the 5-HT2A knockout mice, Sheetal for all the amazing work of handling our animal house. This is where my lab is based. It's actually called the Hutment Lab. It doesn't even show up on this figure because it's a little tiny little hut right on the sea. And I'm very privileged to work with an amazing team of people here. And I'm done. I'm happy to take questions.
Thank you so much, Professor Vaidya, for such a stimulating talk. Fantastic. And honestly, this is great, because this is how science should be done, like without a hypothesis-driven study, but just stumbling upon building blocks and then going from there. We have a lot of questions, actually, so I will take the most pertinent ones first in the interest of time. Shankolpo asks here, so in the presence of very high serotonin concentration and, let's say, toxic amounts, do we observe mitochondrial biogenesis dependent phenotypes?
So if you get a really high concentrations of serotonin, you'll kill the neurons. So there is a, anything is a trophic factor at some dose, and at some dose it becomes toxic. And that's absolutely also true for serotonin. So you go to very high millimolar concentrations of serotonin, the 200 millimolar sort of range, and you will end up killing, neurons are dead. So at that range, I think you lose any mitochondrial biogenesis like function. You're actually triggering death-related pathways and damage. And that's possibly through calcium-mediated toxicity, but it's not clear exactly why neurons are dying. But there is a dose range in which serotonin will also kill neurons.
Right, right. And sort of related to that, Shongram Roy asks, if we increase serotonin level, does receptor levels also increase in the neurons?
So they can, and in more often than not, serotonin would actually downregulate receptor levels. And that's a very classical agonist induced. If you have too much of the ligand, you turn down the receptor so that you have a way of compensating. That's generally true for most receptors. The 2A receptor is an exception to that rule, which actually gets upregulated. So it's a strange receptor from that perspective. It doesn't follow the rule in a sense. But yes, you will change receptor composition if you sustainably keep providing serotonin constantly to neurons. Yeah.
I see. Another question from Anushka Banerjee. She says, does mitochondrial dysfunction then have an effect on serotonin release?
That's a great question. I don't think anyone's ever really looked at it. I mean, that would have to be in the raphe neurons of the midbrain. And one would speculate that if you had mitochondrial dysfunction, you may impact serotonin release. But I can't think of a study which is directly actually assessed whether you can compromise mitochondrial function mitochondrial functions, and as a consequence, directly in study that's actually done that precise experiment, but there'd be strong reason to believe that that is true. It goes haywire, for example, in Parkinson's models, where there is substantial mitochondrial dysfunction and serotonin release is also compromised. So one would guess that it would.
Right.
Yeah.
And another question from Shongram again, actually. So does serotonin have any role for neural development in the growing human embryo? I'm guessing yes, but maybe you can expand a bit on it. Yes.
Oh yeah, absolutely, dramatically. I mean, it modulates progenitor turnover, it modulates migration, it has dramatic influences on pruning and reorganization of circuits. Absolutely critical during the early developmental window for the fine-tuning of multiple sensory systems, visual, somatosensory, etc. So yeah, profound effects in development.
Yeah. I'll sort of take advantage of my moderator rights here and ask a question myself. So you did this in mice as well, and doing these chemogenetic experiments where you sort of induce serotonin stimulation, basically. And do you see any behavioral effects in these mice, or how do you, like, is it normal behavior or not?
Yeah, so if you induce a release of serotonin substantially, you would predict, at least under chronic release of serotonin, you would predict that you would get a reduction in anxiety-like behavior, etc. Those are experiments that are actually directly underway right now using this tool that we have. We haven't extensively profiled the behavior using this tool, but with other models where we elevate serotonin, acute elevation actually increases anxiety, and chronic sustained elevation will give you a reduction in anxiety and a reduction in despair-like behavior. So timing and for how long you're increasing serotonin matters hugely. But I can't give you the chemogenetic answer yet. We're in the midst of doing those experiments. So.
Okay. Fantastic. Thank you so much, Dr. Vaidya. There's another question actually from Shanchari Gorai. She says, for Parkinson's disease, for example, can we use serotonin for preventing further progression of the disease?
It's a great question. It's a tempting thing to start speculating that for neurodegenerative diseases, serotonin may become an interesting potential target. I mean, I wonder if it would, but the question becomes, which class of neurons are you attempting to protect? So in the case of the Parkinsonian model, you're protecting nigra neurons. And I don't know if the same effects that we see on neocortical neurons also hold true at the level of the nigra. And I think there's going to be differential effects based on the cell type, based on what receptors express, because we didn't see it, for example, in hippocampal neurons at all. So this is something that's the jury's out. Until we do those experiments, we won't really know. Yeah.
But also just a small clarification there. So hippocampal neurons have these 5-HT2A receptors and still don't show an increase in mitochondria?
They don't have the 2A at high levels, but most of the 2A in the hippocampus is on inhibitory neurons, on the parvalbumin and somatostatin positive inhibitor. These are the principal neurons that are abundant in the cortex with 2A, but in the hippocampus, the principal glutamatergic neurons do not have the 2A at any reasonably decent level. Very, very, very low levels of 2A expression. So yeah.
I see. Thank you so much, ma'am, for this wonderful session and for answering questions so well and so with so much enthusiasm. Moving on, we have our next speaker, who is Dr. Vatsala Thirumalai from the National Center for Biological Sciences. Please give me a few seconds so I can find her actually. Yes, perfect. Welcome to this platform, ma'am. For the audience, Dr. Vatsala Thirumalai is going to talk to us about fishing expeditions in the brain. She has graduated from Anna University, Chennai with a BTech in biotechnology and has pursued her PhD in neuroscience in the lab of Professor Eve Marder, who probably needs no introduction in the neuroscience community, from Brandeis University, and completed her postdoctoral fellowships in the lab of Professor Klein at the Cold Spring Harbor Laboratory and Dr. Michael J. O'Donovan's lab at the National Institute of Health, Bethesda. She's currently at the National Center for Biological Sciences, Bangalore. She was awarded the prestigious Shanti Swarup Bhatnagar Prize for her accomplishments in biological research in 2020. So congratulations on that. Her science research outreach initiatives are also endless and also include talks at the Blisk Science Cafe, a set of informal science talks organized by the Bangalore Life Science Cluster, and many other science outreach endeavors that she's been part of. Vatsala's research focuses on the development and functioning of the neural circuitry that is responsible for locomotion and motor control in zebrafish. A very, very warm welcome to you, Dr. Thirumalai, and over to you, ma'am.
Thank you, Shreya, and thank you all of the organizers. It's such a pleasure to be participating in a conference that's organized by early career researchers. I would like to tell you that Veronica would have been proud. And it's an especially great honor for me to be speaking in a session named after her and right after Vidita. So, I mean, the TIFR NCBS clusters have such inspiring role models, women scientists, that I truly look up to them for, inspiration every day. Although I didn't have the pleasure of interacting very much with Veronica, she passed away few days before I joined NCBS. But she used to write such funny and quirky emails inviting everybody to come to the student talks. She was so passionate about making sure that the students receive mentorship and training, and they had someone to talk to on campus. That she was truly missed when she passed away. So what I would like to do today is talk about my research, which has been growing over the last 10 years since I started at NCBS. And okay, are you able to see my screen?
Yes.
Okay, so what I'm talking about today is some of our recent experiments using zebrafish as a model organism to understand how organisms move. So the nervous system is actually like the universe, because in the universe you see, starting from, let's say, atoms all the way up to galaxies, we see a range of spatial scales over which there is organization. Just like that, the brain also has organization ranging from the atomic scale or molecular scale all the way up to behavioral scale, which can span even several thousands of kilometers if you think about migrating birds, for instance, right? But at every step, there is a level of organization which is important for the function. For example, if you think about molecules, you have proteins and amino acids and modulators like Vidita spoke about. And then those molecules are organized into signaling networks, like the G protein coupled receptor pathways and other receptor signaling pathways, mitochondria, for instance, that Vidita spoke about. And these mitochondria, for example, can be thought of as cellular compartments. And not just mitochondria, you have synaptic compartments, you have axonal terminals, you have vesicles in those axonal terminals. And then out of those compartments, you make a neuron, which is a functional unit of the nervous system. And the neurons themselves can be connected up in precise synaptic pathways to make neuronal circuits. Circuits are there within our brain. Circuits are also there in the spinal cord. And then the spinal cord can send very long axons down to all of our muscles. And when signals pass through these nerves to the muscles, then you get muscular contraction leading to movement, right? And then the circuits themselves are organized into systems, such as circuits responsible for vision are visual system, circuits responsible for movement are motor systems, and so on and so forth. But a fundamental challenge in neuroscience is to be able to take knowledge that's gained at any one particular spatial scale and then be able to translate it into some sort of meaning at another level, right? And that is because, the experimental systems that are well suited for analysis at a certain level, let's say a molecular scale analysis or so on, may not be suitable for analysis at a different level. If you're looking at circuits or behavior, the kinds of experimental systems that you might use are very different from what you might do if you're looking at a molecular scale. And so we are always in a quest for getting a model system in which you can get an integrative understanding, where you can go from one level of analysis to another. And that's where our favorite model organism, the zebrafish, comes about. You have, these are fish that are endemic to India. The fish on top is a female fish and the fish in the bottom is a male fish. And the reason for choosing this as our model organism are several fold. Number one, they have a very short lifespan, so you can study different developmental stages quite quickly. And the fertilization happens externally instead of in utero. So you have access to the embryo from the very first single cell stage all the way up to the adult. And most importantly, they are transparent. And in the early larval stages, they don't have a skeletal system, so it's easy to access the brain. And because they are transparent, you can just put them under a microscope. And if you have ways of genetically engineering the neurons to express fluorescent proteins like GFP over here, then you can observe those neurons, both their structure as well as their function, and ask what is going on when the animal is trying to move. And you can record from single neurons and populations of neurons using electrophysiology. So given these advantages of the model organism, we wanted to use this organism to ask, how does the brain generate movement? And recently, I'll tell you a story that we've recently completed, which has to do with control of speed during locomotion. And this work was done by my student, Urvashi Jha, wherein we asked, what are the signaling mechanisms? What are the neurons doing when an animal changes speed, and why does it change speed? So why do organisms need to change speed? As we all know, in the wild, we need to change speed to be able to find food in the case of a predator, or if it's a prey, then the prey needs to be running very fast to be escaping the predator. So both for survival and escape from predators, as well as survival in terms of finding food, control of speed is necessary. Then, of course, is the need for finding romantic partners. And I thought I'd use this example carefully.
I think it froze a little bit. If you can repeat again, or can you hear me, Dr. Thirumalai? I think it froze. Maybe if you go back to the previous slide and please bear with us. We're having a small technical glitch and we will be right back. Just give us a few moments. I think we lost Professor Thirumalai and think she will be back. Please bear with us. I think Professor Thirumalai is back, so. We're extremely sorry for the inconvenience. Please stay with us, everyone. We're just going to be back on track in a few seconds. And there we go.
Can everybody see the screen?
Yes, ma'am.
Okay, so sorry, my computer just crashed, so that's why I'm not turning on my video, just so that we keep things light. So I was saying that there is a need to regulate speed, and the trade-offs for regulating speed are that within biological constraints, which are imposed by the muscle mass that a certain organism has, within those biological constraints and the amount of energy that is available, the speed has to be carefully regulated for the behavioral demand that's present at that moment. So for every moment, the organism needs to carefully evaluate, how fast should I be moving and how much should I be moving, so that the energy expenditure is optimized. So how do organisms do that is the central question that we wanted to understand. And zebrafish larvae are known to swim over a wide range of speeds, whether they are just exploring the environment, like this zebrafish larva over here, just swimming in the dish, in no particular direction and in no set specific speed, or when it's engaged in a reflex behavior like this larva over here. We've triggered a reflex known as the optomotor reflex, which is the ability of a larva to follow black and white stripes. And the larvae do this because they are normally living in slowly moving water. And when they sense that the water is receding from them by means of optic flow, then they swim to keep up with the water flow. So when they see these black and white stripes moving in the forward direction, they are given to believe that the water is pushing them backwards, and therefore they swim in the forward direction, and this is known as the optomotor response. And this is what we used as a mechanism for inducing various speeds, because as you can see in this figure, this video, the larva is roughly swimming at the same speed as the grating speed. So you can change the speed of the larva by changing the speed of the grating. They also have very fast behaviors like the escape response happening over a millisecond time scale. Now, what we know about speed regulation over the last 50 years of research is that the speed is set primarily by circuits within the brain. So when an organism decides to move, the action plan for the movement is actually decided by various regions of the brain, such as the cortex, the basal ganglia, and the cerebellum. And once the motor command is made, it is sent to a region of the midbrain, where the frequency with which the limbs have to move, whether it's the tail movement in fish for swimming or the wing beats of an insect, not an insect, sorry, of wing beats of a bird, or let's say the limb movements of a mouse, all of those frequencies are set by this midbrain region. In insects, it's set by various ventral circuits present in the ventral nerve cord. Now, the frequency of the movement can be changed, and if the frequency of the limb movement is changed, then the organism moves faster. Once the midbrain decides on a particular frequency of movement, then it engages gears just like you would in an automobile. Faster movements, faster frequencies engage faster gears, and therefore the organism moves faster. Now, finally, after all of this, the command goes to a type of neuron called as the motor neuron. These neurons are present in the spinal cord, and their axons are what innervate the muscles causing muscle contraction. So the final command from the nervous system is taken by these motor neuronal axons. Okay, so that is what we know. But what is unclear in this whole scheme of things is the role of neuromodulation, neuromodulation like serotonin, which you already heard about, or other chemicals such as dopamine, acetylcholine, or norepinephrine. The reason why this is important is that we know that these chemicals are capable of reconfiguring circuits, changing them in fundamental ways. So the question is, you have this very precise set mechanism of cortex and basal ganglia talking to midbrain, and then the midbrain talking to the spinal cord. But what happens when these circuit properties are changed by neuromodulators? So that is the question that we wanted to understand. And we knew that dopamine is present within the spinal cord. So we chose to turn to dopamine using drugs that affect dopaminergic receptors, D1-like receptor in this case. And what we found was in fact very surprising. We found that in normal water, if the fish swam at a certain speed, I'm sorry, the video is not playing in here. But what we found is that the fish swam at a particular speed in normal water, shown in black dots over here. But when we activated dopamine-like receptors, they swam much faster. And the opposite was true when we blocked the dopamine receptors, then the speed came down. So what happens? Why do larvae swim faster when they are challenged with dopaminergic drugs? And we can answer that by doing a simple experiment where we embed the fish larva in a jelly-like substance, and then we show them the same black and white gratings, and we look at the tail movements. This is done so that you can put it under a microscope and you can measure the tail beat kinematics at a very fine scale. And what we found was that the larval tail bent over a larger extent when we applied dopaminergic drugs to them. So the larger tail bend resulted in greater propulsion in the forward direction and therefore faster swim beats. So that is what is shown here. With the blocker, the opposite was true. The tail bend amplitude actually went down. So what makes the tail bend more? Is it that there is greater muscular contraction, or is there a nervous system component to it? To understand that, we paralyzed the larva and then we recorded the nervous system signals coming out from the motor neuronal axons and asked how do these signals look in control versus in dopamine. And what we found was that the signals looked much bigger when dopaminergic agonist was bath applied, as shown here, without any change in the frequency. So the frequency of the tail beat remained the same, but the amplitude increased, right? So that means that the brain is actually setting the frequency at some level, frequency of motion, but that for the same frequency of motion, the larva is able to swim faster, and this was novel. So what explains this? So to analyze this at the next level, what we did was the same preparation where it's paralyzed, but this time we are recording from single motor neurons and we are asking what happens to the action potentials being generated by these single motor neurons. So this is in control. The beige colored bar represents time when the grating was moving. The white bits are times when the grating was not moving. So the larva only elicits swimming when the grating is moving. And you can see that in the presence of the agonist, when the grating is moving specifically, then this motor neuron generates more action potentials. These tall signals are the action potentials generated by the motor neuron. And when the dopamine receptors are blocked, then those action potentials go away. So this is consistent with saying that the dopamine is making the motor neurons fire more, be active more. So, of course, the next question is, how is it the motor neurons elicit more action potentials? Now, every neuron is like a little electrical device. You put in current into the neuron and it generates a voltage output in the form of action potentials. So to recreate this in the experimental system, we injected steps of current in control and in the presence of the dopaminergic drug. And we found that when dopamine is present, for the same amount of current injected, the neuron is able to elicit many more action potential. So it's just more excitable. So that explains it, that it receives the same drive, but for that same drive, it's able to produce more action potentials. And because there are more action potentials, the muscle contracts more. And because the muscle contracts more, the tail bends over a larger extent. And because the tail bends over a larger extent, the larva pushes forward to a greater distance. And because of that, it's able to swim faster, right? So going from a cellular level all the way to the behavioral level. And that's not the complete story though, because we also found evidence that the drive coming into the neuron is also increased on top of the change in the excitability, right? So both of these things put together make the neuron fire more. There is increased excitability, firing more action potentials, and there is increased synaptic excitation coming into the neuron, adding together, making the neuron fire more. And at a population level, if you're looking at all motor neurons, in control, there are very few motor neurons which are participating in this behavior. But when dopamine is activated, then many more neurons, especially motor neurons, which typically fire only during fast swims, are now recruited, and they are all firing action potentials, making the fish swim faster, right? So to summarize that, then you have normally brain commands telling the spinal cord to generate locomotor rhythms of a particular frequency. And in the presence of neuromodulators like dopamine, then the same swim command is taken, but the firing rate of motor neurons is increased. And because of that, you get a faster locomotor rhythm. So this is like a parallel or alternate pathway to regulate speed, which is different from the canonical model where the speed is altered by changing the frequency of the locomotor command. So that is the end of this story. I'd like to thank my group, which is shown here, and also all of the funding agencies which helped us generously over the years to conduct this research. And I'll be happy to take questions.
Thank you so much, Dr. Thirumalai. You can post questions on the box. I think there is the first question from Debayon, who's also part of our BioSpeak team. So he says, does butyryl cholinesterase play a role in neural modulation and neural disorders? Ma'am, if you could switch your video on, then maybe we could see you as well.
Yeah, sorry. Yeah, I had turned it off during my talk. I'm very sorry my computer crashed during the talk.
No, this can happen. It's okay.
So I'm not familiar with butyryl cholinesterase. There is acetylcholinesterase, which is present at the neuromuscular junction cleft. And of course, it has a role to play with speed and muscular contraction, because the acetylcholine that's released at the terminal is degraded by this enzyme, and the increased or decreased activity of this enzyme has a crucial role to play in the availability of acetylcholine, how long it's available at the cleft for the muscular contraction to take place.
Thank you. And the second question from Shweta Bhagwat, it's more of a general question. So she's asking, this is something very similar to chemotaxis. I'm guessing she means implants. Do you see any correlation with the mechanism here in this case?
Yeah, so unlike a goal-directed behavior, for example, tracing a olfactory cue or tracing a gustatory cue, this is a reflex response, like I said. So it's set by the frequency of the visual motion, not just bars. You can pose any sort of visual scenery, project it onto the fish, and depending on the velocity with which the visual information is flowing past the retina, the larva will generate swims. But chemotaxis is also present. Fish have a wide range of olfactory behaviors, both aversive as well as attractive. And that is something that a lot of people around the world are actually investigating.
I'd like to ask a question myself again. So this, for instance, we know that there's the basal ganglia system in the vertebrate system where D1 promotes movement and D2 inhibits, or the other way around actually. Is there something like this in the zebrafish system as well? Is this whole thing a complementary system of the basal ganglia and vertebrates, let's say?
So fish have a basal ganglia homologous circuit. It's not as well defined as it is in the mammals, but they do have homologous circuits based on mRNA expression of markers. People think that the telencephalon of the fish brain does indeed have centers which correspond to the mammalian basal ganglia. Again, these are quite early days and people haven't worked out the functioning of these circuits. The telencephalon of the zebrafish is actually, wilderness. No, not many people work on it. There are very few groups such as Hitoshi Okamoto in Japan who've done studies on it. But other than that, it's something that worth exploring, that's out there, that for people to follow up if they are interested in.
Thank you so much, ma'am. We have a few more questions. Manish Mandhotra asks, is zebrafish being used to study cancer? Please explain, and also for studying neurological disorders.
Absolutely, absolutely. See, one of the fundamental advantages of using the zebrafish is that because it's a vertebrate, the organization of the body as well as the developmental stages are quite conserved between mammals and zebrafish. Many early events of development are conserved, like the gastrulation, layers formation, organogenesis, somite formation, etc., etc. So it's primarily it started out being used as a model for developmental biology. But because of that, now we know a lot about homologous genes between zebrafish and mammals, looking at genetic pathways. And when you talk about genetic pathways and you're interested in gene function, it's quite useful to put it in the zebrafish model organism and then ask what happens when this gene is mutated or that gene is mutated. And that is how it's being used as a model organism for studying not only cancer, but also neurodegeneration and many other disorders, asking, what is the function of this gene or that gene, or this network and that network, what kind of small molecules can modulate this process. For example, this is a huge area of pharmaceutical research, where if you have a library of compounds, you can use zebrafish as a preliminary screening model system to ask what is the effect of this small molecule on a particular cellular process or cancer or some sort of disease, and so on and so forth. Yeah, that's being done in a lot of different places.
I see. That's very good to know, actually. We don't have really so much knowledge about zebrafish research somehow. I will take two last questions in the interest of time. One is actually from Professor Vaidya, who's just spoken on our platform before. So she says, once a behavior pattern is strongly reinforced as rewarding, does D1 receptor stimulation still enhance speed? Or does it go independent once the reinforcement learning has kicked in fully?
That's a very interesting question, very interesting. But this is something that we would like to look at. We've just started looking at learned behaviors in zebrafish. We don't know. The short answer to Vidita's question is that we don't know. And this is something that can be analyzed in zebrafish because we have access to the whole nervous system and the whole organism while it is behaving. And that is something that we are following up in the lab right now.
Thank you for the response. The last question is from Debayan Sharkar. He's also asking about, he says, a very interesting talk. Thank you, Dr. Thirumalai. But do you think the dopaminergic activation eliciting higher firing frequency of the neurons is related to higher motivation and response, which means this will work with other stimuli other than light grids?
Right. Again, a fantastic question. And this is something that we grapple with when we think about fish. I mean, what is motivation in a fish? When do you call the fish to be, I mean, we try to impose anthropogenic words to anthropomorphic words to fish, right? And it's interesting to think about, but it's also a very difficult question to ask. But as he says, this is again an experiment that we are following up in the lab to ask. We haven't looked at other sensory modalities, but we are looking at other triggers of locomotion, so to speak, such as the escape response that we talked about. Is it that dopaminergic modulation affects only optomotor response, or is it that it affects a range of other locomotor responses? So we are looking at escape response, for example. We're looking at spontaneous exploratory behaviors. We're looking at prey capture. All of those things can be looked at. But looking at other modalities would also be other sensory modalities that trigger locomotion or learned behaviors, like Vidita pointed out. All of those are open questions to look at.
Thank you so much, Dr. Thirumalai, for this wonderful talk and also answering the questions with so much enthusiasm and interest. It was a delight to have you on the platform, and we wish to do more things like this with you again.
Thank you. Thank you so much. I really had pleasure giving the talk and answering so many questions, interesting questions. And thank you once again. I mean, it's remarkable that young scientists like you are taking the initiative to organize a conference like this. So good job, and keep it up.
Thank you so much for those encouraging words. With that, we move on to the next session, which is, let's see if we can find Dr. Uma Ramakrishnan. Yes, so this session was actually started yesterday. It's the Jagannathan Vijaya session on biodiversity and animal behavior. For those of you who are joining us today for the first time, Jagannathan Vijaya was born in 1959, was India's first female herpetologist, and was known as the Turtle Girl for her immense contribution to the conservation of turtles. She documented the movement of turtles all over the country and worked as an assistant to Edward Moll, who was the chairman of the World Conservation Union's Freshwater Chelonian Specialist Group, and on his nationwide surveys. J. Vijaya, or VG, as she was fondly called, was born in Bangalore in 1959. Since childhood, she was an avid admirer of nature, grew up idolizing the likes of Jane Goodall, Jacques Cousteau, Diane Fossey. She started working with Edward Moll at the age of 22 and documented the entire trade line of certain species of turtles from their capture, from their habitat till the transport and selling in the meat markets. In honor of India's first female herpetologist, Biologically Speaking is proud to present the Jagannathan Vijaya session on biodiversity and animal behavior. Two speakers, Dr. Anindita Bhatro and Dr. T.N.C. Vidya, were featured on this session yesterday. Today, we have Dr. Uma Ramakrishnan from NCBS Bangalore joining us on HerStory of Science. Please welcome her. Dr. Uma Ramakrishnan is an Indian molecular ecologist and an assistant professor at the National Center for Biological Sciences in Bangalore. She studies population genetics and the evolutionary history of mammals in the Indian subcontinent. Dr. Ramakrishnan did a Bachelor of Science in Physics, Chemistry, and Mathematics and a Master's in Biotechnology. And she subsequently received a PhD from University of California, San Diego, following which she was a postdoc at Stanford University. Her research focuses on revealing the processes that drive patterns of mammalian genetic variation in the present and the past. She uses field-collected samples, assembles molecular genetic data, and analyzes these data with phylogenetic, phylogeographic, and population genetic inferences. Her lab developed methods to conduct population monitoring and landscape population genetics with tiger fecal samples. Professor Ramakrishnan has collaborated with the tiger expert and former director of Wildlife Conservation Society, India, K. Ullas Karanth. Together with Karanth, Professor Ramakrishnan's lab used genetic sampling to estimate the tiger population in Bandipur National Park. Her research on the connectivity of tiger populations in central India was presented in court to stay the widening of the NH7, which cuts across the Kanha-Pench corridor. She was awarded the Parker Gentry Award for her work in tiger conservation in 2016. And in 2019, she was elected as a fellow to the Indian National Science Academy. Please join me in welcoming Professor Uma Ramakrishnan from NCBS Bangalore to this platform today. Over to you, ma'am.
Hi, thanks so much, Shreya, for that very long and a little embarrassing introduction. So wonderful to be here with all of you today. And I'm sorry I couldn't join you yesterday. The reason I'm looking a little tired maybe is because I actually just came back yesterday from this fabulous place. I was traveling yesterday and I'm now in quarantine. But I'll just go ahead and share my screen and let me start. Yeah, so I was over here. This is Kaziranga Tiger Reserve, and it's a beautiful place. It's in the floodplains of the Brahmaputra. Every year, a large proportion of this protected area is flooded. It's an amazing landscape and amazing to even try and imagine how people and animals live here, running away from water at all times. While we were there, we saw, the Assam state animal, the rhino. Really a nice picture here of a baby rhino as well as some adults. And these are very bad pictures. I'm not a photographer. This is just from my iPhone. The critically endangered slender-billed vulture, which was preying on a scavenging on a close-by wild buffalo kill. And of course, charismatic elephants, really big megafauna, right, which live here. What we were actually trying to find was this, and this is a pretty bad video, but hopefully you can see that it's a tiger. Oops. I hope you can see it looks a little odd. So it's actually a different tiger. It's this golden tiger. And it looks very bleached out, right? It looks very different from a regular, orange or a wild type tiger. Excuse me. So a lot is known about color variation in tigers, and there have been studies based on tigers in captive facilities in the US, which have identified mutations which are responsible for, the white tiger and the one we just, I showed you a video of in Kaziranga. We didn't see it. I was just there for 5 days, the golden tiger, but it's known now from several evidence from tourist pictures, from camera trap data, and from sightings that the golden tiger, indeed, there are golden individuals in Simlipal, at least one, okay? And this is the snow white tiger, which is a double mutant of golden and white. So what we were hoping to do, of course, is to, we work with fecal samples, as was just mentioned in my introduction, try explaining that to anyone. But we basically walked through the forest. In this case, we were in the Jeep because of danger from being charged by rhinos, and we were charged, so this is a real danger. And collect fecal samples, and hopefully one amongst the fecal samples we'll collect will have this mutation which has been identified earlier, from some other study for the golden tiger. But this is a case where phenotypes, the genetic basis of a phenotype is known. But recently we've completed some work, it's still in revision, where we've looked at scenarios where there are these other really beautiful tigers. This is from a picture from Simlipal of a pseudo-melanistic tiger. It's pseudo-melanistic because it's not completely black, as the term melanistic would imply. But this is kind of a blackish tiger. And my student Vinay, who's done an exceptional job and is also now in Assam, in Kaziranga, sampling for the golden tiger, was able to look at this and try to first of all understand what the mutation is which determines this phenotype. And we got hints from these earlier studies on cats, which have basically identified that, in a regular cat which has stripes, there are basically this particular gene called TACPEP is normal, but association studies showed that in these type of cats where they don't have stripes, they have these whorls, you have a mutant version of the TACPEP gene. So this mutation in the TACPEP gene results in, the disruption of striping as a pattern, right? And so you get this kind of, funky looking cat. Luckily for us, in the Nandankanan Zoo in Odisha, there was a litter born which had basically a white mother, a white but pseudomelanistic daughter, and an orange pseudomelanistic son. And we were then able to use this pedigree and genome sequences from individuals in this pedigree to identify the genetic basis for this phenotype of the pseudomelanistic tiger. And what we found is that there's this one single point mutation, which is in the zinc binding domain, and it's a missense mutation. So it changes the protein, and it's a very conserved region, suggesting that it probably, we would infer that it renders the protein to not have function. So while the mutation is not the same for as in cats, tigers also seem to have mutations in this gene causing this phenotype. So this is all like genetics, right? So what what's actually happening here? What's also really interesting then is that when you look at the frequency of this mutation, this pseudomelanistic mutation, we looked for this mutation genetically, because you may not see a pseudomelanistic tiger because it's a recessive trait, and an individual has to have 2 copies of this trait to look pseudomelanistic. So we looked at 400 tigers across tiger range, mostly in India, because this is where it has been found, and found this allele only in the natural habitat of Simlipal Tiger Reserve and 2 zoos, one in Nandankanan, which I just showed you, and also in Chennai Zoo, right? And what was very interesting is that in Simlipal Tiger Reserve, the frequency of this allele was almost 60%, whereas outside of Simlipal Tiger Reserve, it was 0. It was just basically not there, right? So, so I guess this is kind of the build up to my talk, and this is how I do research. So I thought it's a nice way to start. I basically do a curiosity-driven research. I just follow my nose if something seems interesting and just see what's going on, try to understand what's going on. So here is a situation where we have these rare phenotypes of tigers. They're rare because they're not found anywhere else, yet in some locations, they might be common, right? For example, if the frequency of the pseudomelanistic allele is 60% in Simlipal, we expect about 30% of the individuals to actually be pseudomelanistic. They would have 2 copies of the recessive. And this basically takes us to the larger question of biodiversity, okay? So we started out with genetics and curious looking individuals, curiosities, almost like vanity tigers. But how does this lead us then to thinking about biodiversity? Well, biodiversity today on our Earth is in crisis. We all know this, and somehow we don't internalize it. We may, but I don't think we internalize it enough. And biodiversity does matter to all of us. The water we drink, the fruits we eat, and the air we breathe are all a product and function of the biodiversity that surrounds us. But that same biodiversity is under threat, and I'm not just talking about the ecosystem and climate change. I'm talking about species. 1 in 4 species of mammals is threatened with extinction, and this is especially something which bothers me a lot, because all of the data so far suggests that from the 1970s, despite actually global engagement with the issue of biodiversity and conservation, species have been in decline. And I was born in the 1970s. So it really hurts almost personally to think about biodiversity loss that is ongoing pretty much during my lifetime. And I hope that will change in all of your lifetimes. I really do hope so. So if you're thinking about this as a scientist, what is it that causes extinction? It's basically, extinction is a function of population size and population isolation, as well as genetic variation. So literally small and isolated populations are more likely to go extinct, and also populations with low genetic variation. The effects of genetic variation on extinction can be seen in two ways. The first is inbreeding depression. When you have small populations, individuals tend to mate with relatives and you have inbreeding, and this could lead to inbreeding depression or the lower chances of survival or lack of fitness or lower fitness of inbred individuals. Additionally, you may have drift or stochastic by chance changes in allele frequencies. This also happens in small populations. And basically, tigers are a very a good example of an endangered species. Over the last 300 years, India has significantly lost green cover, and today you can see the patches of green are very small, and so the number of individuals now which are in these green spaces obviously are then isolated from each other. They've also seen extinctions of several subspecies like the Caspian tiger, the Bali tiger, the Javan tiger, and the South China tiger. And as you know, there has been significant hunting and continued trade for tiger parts. So the pressures for tigers in terms of population size reduction are still ongoing, despite extensive protection on the part, even for example, of the government of India. So we basically study tigers through a genetic lens, and we use genetic data as a tool in some cases to understand ecological parameters like population size and gene flow or connectivity. And then more recently, we have also been looking at processes like inbreeding and drift in small populations. And we call ourselves molecular scatologists. We sample tiger DNA in terms of fecal samples, bring them to the lab, bring this back to the lab, and then try to work wonders, massage data out of these really poor quality biological samples. Some work we did, as was mentioned in the introduction, in central India, was helped to reveal that high traffic roads and high density human settlements are barriers for tiger movement. And we did this using genetic samples collected from across the central Indian landscape, walking thousands of kilometers. And basically, we also did future projections of landscapes to show what types of populations and what types of future scenarios would cause high or low extinction. And basically, we again tested and showed that small populations or small protected areas in central India, which are isolated, are much more likely to go extinct, whereas large and connected protected areas are less likely to go extinct. But there's a bunch of protected areas in the middle, which you could either potentially increase population size or gene flow and change their fates, flip their fates from extinction to continued survival. And so basically these types of simulations showed, for example, that this was basically the corridor between Kanha and Pench, NH7, which is being extended in the introduction was mentioned a stay on the highway that was overturned, and the highway construction started. And then we were able to, not we, NGOs on our behalf were able to argue for that extending this highway would disrupt a natural process of connectivity and cause increased extinction. So our models, for example, showed that extinction would increase by 50% should this highway be widened and become a barrier. And this was used then by the court to order the National Highway Authority of India to put in this 700-meter long underpass so that the animals could actually keep connectivity intact. So this was very nice to actually see that these models and data and models based on data could actually be used for some action on the ground. We've also been working in Ranthambore, and we've been looking, again, this is an isolated population, and we've been doing a lot of genomics to show that, these individuals are inbred. It is a small and isolated population, as I said. And tigers here are twice as inbred as others in any other part of India. What's also interesting is that we can predict based on studies of mutations of other species, what we understand about proteins. We can actually predict what mutations or changes may be deleterious or bad alleles and look at mutation load. And this shows, for example, that mutation load, or the proportion of deleterious or bad mutations, is high in Ranthambore tigers, as well as close to fixation in many cases, which is high in frequency. Not just the numbers are high, but many of these mutations are close to fixation. So this brings me back to the story of Simlipal, which I left half done. I just told you about how we found the mutation. What I didn't tell you about, not that we know the answer for sure, is why is the frequency of this mutation so high in Simlipal. So Simlipal is also a very small population, in fact, much smaller than Ranthambore. There's between 10 and 20 tigers in Simlipal. Our genetic data suggests two things. One, that Simlipal is isolated, and I'm not showing that to you here. But what I am showing you is relatedness between individuals, tigers in Simlipal versus other protected areas. And you can see, for example, that, on an average, tigers in central India are not very related. Some are, they go up, the relatedness does go high. But on an average, tigers in Simlipal are quite related to each other, almost on the order of, half Sims or something like that. Not as bad as Ranthambore, but still, on an average, tigers are quite highly related to each other, suggestive of them inbreeding. Additionally, when we do simulations, again, to ask what might allow the frequency of this allele, which is near absent or close to zero outside of Simlipal, to increase to a frequency of 60% in Simlipal, the most likely scenarios are what are called founder effect events, where a few individuals survived and maybe two individuals or three individuals, and one among them must have had the allele, right? Must have had this mutant melanistic, pseudomelanistic allele, and only that way can the frequency increase to the substantially high, frequency we have today, we observe today. Okay, so basically, this is also interesting because, I don't know whether you've seen the news, but the last 7 days, there have been massive forest fires in Simlipal. And these types of stochastic events could actually cause, a loss of vegetation, die-offs among the ungulates. And when the prey base reduces, tiger populations will also be affected as carnivores. There's also been known records of poisoning of the salt licks and basically, again, die-offs of prey. So it's possible that Simlipal tigers have been through these kinds of bottlenecks. And given that they are isolated with no other input of genetic material, this seems to be the most likely explanation for why there's a high frequency of this allele here. So to summarize, tigers are a classic example of an endangered species. And they have lost a lot of habitat. You can already see that populations are relatively isolated, some of them. Some populations are still connected, like in central India, but the way we manage the land outside the protected areas could change this in the future. So it's critical for us to be cognizant or to know about what impacts our actions are going to have, not just on ourselves, but also on biodiversity that is around us. And already genetic changes and effects of isolation are apparent, with rare phenotypes becoming common in certain places. Whether this is the story of the golden tiger in Kaziranga, that will take a few more years to figure out, but hopefully we'll have more on that soon. So of course, I am lucky to work with an incredibly talented group of students like yourselves, and also lucky to have many people who fund these adventures of ours. So thanks very much, and I'll stop there. I hope I didn't take too much time.
No, this is perfect. Thank you so much, Professor Ramakrishnan. We have lots of questions actually. For the sake of time, we will first take the pertinent questions. So Professor Vaidya actually asks, is the introduction of tigers from other locations, will it help to introduce greater genetic diversity? Is that a viable strategy? But also, is there a danger of a tipping point after which even that would not be a viable strategy? And I ask this in conjunction with another question from Shonchari Gorai, who says, we know that the Asiatic cheetah has been extinct from India. And is there a project to bring these cheetahs from Iran to India? If it's at all possible, what is the effect of the environment? Because it would be a different environmental landscape.
Yeah.
And if you could comment on that.
Sure. Thanks. Those are both great questions. And I think that they basically speak to, in some sense, when we think about conservation or conservation action, a lot of it is based on ecology, about numbers and so on. Or when you think about the cheetah, it's about, okay, how can you bring cheetahs from Iran? What we are not considering are who are those cheetahs? What is their genetic makeup? Or if we are going to have to rescue populations, whom are we going to rescue them with? Are we just going to catch a random tiger and move it, which is what is usually the case? Problem individuals are just translocated. Or are we going to do this on a scientific basis to try and, initially think of how much gene flow is important, what will be the impact of gene flow and so on. So I think that, yes, definitely. Unfortunately, I think that in the future, management and movement of animals will be the only way forward, because we may lose natural connectivity. I hope we won't. Of course, we should push for natural connectivity as long as we can.
Right.
But in the event that we lose it, then it will have to be the only option. So, for example, with cheetah reintroduction too, I would say it would be really important to think about who those cheetahs are. Are there unique adaptations of Iranian cheetahs which are different from those in Africa? What might the effect of that be here, and so on? So I think there's a lot of work cut out for geneticists, many questions which maybe we can't answer, but at least we should start trying.
Right. And do you think there's a danger of a tipping point as well?
Sorry, of what?
The danger of a tipping point after which it's not. Yeah,
so there is, right? So basically that's what's happened, for example, in cases like the Tasmanian devil or the California condor, where basically now we've resorted to captive breeding or introducing favorable alleles in the case of the Tasmanian devil, which has this mouth cancer, contagious mouth cancer. And very recently there have been 2 endangered species which have been cloned. That's like the super management approach. The Przewalski's horse and the black-footed ferret have been cloned using biobank cells, which introduce genetic variation into the current population, which is now extinct. But I really hope we don't have to go there, but yeah.
Right. So on the same note, actually, so Sanjana from our team, actually, she's asking how feasible are wildlife corridors in areas of high urbanization? Is that a strategy?
Yeah. So the thing is that, for example, wildlife corridor means how do you know where the animals will move? So in places like the US and Canada, it's all fenced, right? And then you're funneling animals. So you're basically fencing out everything else. This is not an option in India. Our protected areas are very small and people use the protected areas and we can't do this. So basically we have to think of ways to attract animals maybe to somehow move. So think about, say, for example, elephants. Elephants love things like jackfruit. Okay. So if you, this is just complete, this is not entirely scientific, but you have to somehow think of a way to pull animals to move in certain routes. This may be possible for some species like maybe bears or elephants, but it would be very difficult for tigers. And there's also a lot of conflict which happens when tigers move through human dominated areas with livestock kills and so on. So yes, maintaining connectivity is very challenging. But for example, in the Northeast, my impression I haven't done enough work there to know for sure, but there is forest cover. There is what is called structural connectivity, the forest or the ability to move is there, but the prey base is very low. So there aren't populations which are robust enough to produce individuals who can move. So I think the problems of connectivity are different in different places. I think, for example, in central India, it would have to be these underpasses and a lot of management of conflict. That's the way the connectivity can be maintained.
That's fascinating, actually. I will take two more questions in the interest of time. So one is from Sundaram, who's also part of our platform. So based on existing genome sequencing data, do you use mathematical modeling to predict the emergence of rare phenotypes and also inbreeding depressions in a particular region?
Yeah, so inbreeding depression, so inbreeding depression is decrease in fitness. Now, uh, one way to study it is using pedigrees. And so that's what we're trying to do now in, say, a place like Ranthambore, is to reconstruct the pedigree based on all of the individuals. We sample known individuals and then we try to ask, who are they related to, and build the pedigree. And then we can actually look at whether inbred individuals have lower reproductive success than outbred individuals, something like that. The other way is what we're doing. It's like a biology free way, like with genomics, where we are predicting what the bad or deleterious alleles might be. And then we can track those alleles to look at whether they have fitness effects. These rare phenotypes themselves, I don't know whether they have fitness effects. But what I was insinuating is that they are associated with inbreeding, because otherwise, since they are only showing up as recessive homozygotes, unless there's inbreeding, they may not be thrown up at all. They would simply be rare and not, not like seen.
Right,
I hope that's clear.
exactly.
Yeah.
The last question is a bit more philosophical, in the sense that, so Manish Malhotra asks, can you please suggest measures that we as biologists can do to contribute in protecting tigers and wildlife in general? And to that, I would like to add, in the sense that, could you elaborate on how difficult it must be to conduct these studies and collect fecal samples over forests and areas, basically?
Okay, so I guess that, I think that as scientists, we should all respect, all kinds of data. And I think it's really nice. I think the community is opening up a lot more now. And there's a lot of different types of scientists who do different kinds of things. And I think as long as we respect that, different people are driven by different things, I think that's really nice. And I think we should all be open to considering, data and evidence. So even as a scientist, whether I'm a neuroscientist or a ecologist, data is the holy grail, right? Evidence. So we should really be, in all walks of life, we should kind of probably support that to the outside world versus impressions. I think that's kind of important. As far as doing the work goes, I guess we are so lucky to be able to work in these spectacular places. And the biggest challenges are logistic and bureaucratic. They are not to do with the field work. I mean, it's really a privilege to be able to walk in a protected area for the whole day and submerge in the sights and the sounds. And I think all of my students would agree to that. And I feel lucky every time I go. So I think that it's mostly that since we work outside, all of our curiosity is outside. It's something which is under the view of a lot of people. Like if I was doing an experiment in my lab, it's just me and the experiment. But whatever I do, we do, is viewed by lots of people. And so there's a lot of scrutiny. So it's something which there's a lot of regulations, a lot of permitting. And that's something which sometimes can be challenging.
That's true. That's true. We don't think of it in that way. But it must be a thrilling adventure to be out there and doing this kind of work. It's very real time.
Yeah. And I must say that what is also for me, and this is again a very personal thing, is I have been so lucky to also in all of these field trips encounter incredibly warm, exceptional, hospitable people from those places. So that is, for me, I have learned so much about India, which I didn't know before.
Right. Thank you so much, Professor Ramakrishnan, for this exhilarating session. It was really great to have you on board, and hope to have you on our platform in the future again.
Thanks for the
Thank you
invitation.
so much.
And yeah, sorry I couldn't make it yesterday, but
Oh, no worries. As long as you're here, it's great.
bye. Bye.
Bye. Thank you, everyone, also for being a terrific audience and for asking questions, very insightful ones. Please join us back at 4 p.m. for the session, the Kamla Sohoni session on gender equity in Indian academia. We are going on a short break now, but we will be back soon, so please stay tuned.